Hi everyone, this video is about DNA replication, and we might possibly throw in some transcription translation stuff at the end, we'll see. Okay, so let's just start out by drawing a bubble, a replication bubble. So we're just gonna draw the framework out before we write anything else in. Oops. Okay, so something to look at first, just like big picture, is let's write this first. DNA replication, obvi. Okay, so we have one good full strand of DNA. And we are going to split that in half. Okay, so we are splitting that in half down the middle here. Sorry, that's not perfectly centered, but you get the point. We're splitting it down the middle, and then each half gets a new half. So the black in this example is our parent strand, is our old strand, and then, sorry, parent strand might not be the coding strand, I think. Sorry, just say this is the old strand, right? The black is the old strand. I'll get back to that. The blue is the new strand. That's all we need to know for now. Okay, so I don't think that he really teaches this next part, but it helps me understand it, and a lot of people I've talked to helps them understand it as well, so I'm going to teach it to you. We have the origin of replication is going to be here and here, and that's in the center of our bubble. Origin of replication. So if we were to look at this, we're starting right in the middle here. So if we have, picture my fingers as DNA. So we are starting in the middle, and then the bubble is forming from the middle. So we start in the middle and we're going out. So we're forming this big replication bubble, and we're opening in both directions. Okay, so we're forming a replication fork here and a replication fork here, starting at this origin of replication. So if that helps you, it helps me to know that that's why we're going both directions, because we started in the middle. So this right here, in general, is our replication fork on both sides, right? Right, okay. So to do this unwinding, we use, draw little triangles here. We use DNA helicase. So DNA helicase is the enzyme that's going to come in and unwind our DNA. So let's label that. DNA helicase. It's going to unwind the double-stranded DNA. Oh my goodness, unwind double-stranded DNA. Okay. As it's doing that, we have topoisomerase. I'm just going to draw a little another enzyme over here and over there because it's on both ends. Topoisomerase is going to unwind the DNA to relieve any stress, any torsional stress. So it's going to uncoil it. Yeah, so topoisomerase is going to unwind DNA and relieve stress on the DNA. Might be important to know that there's topoisomerase 1 and topoisomerase 2. Topoisomerase 1 would cut one strand of the DNA to unwind it and then glue that one strand back together. Topoisomerase 2 would cut both the strands of the DNA to unloop the DNA, further down, and then it would put it back together. So there's two types of topoisomerases to keep track of. Also, as we're unwinding the DNA, we have single-stranded binding proteins. So I'm just going to draw some here and here. So single-stranded binding proteins. And those are going to hold the DNA open because we're unwinding it. But before we add down any new DNA, it's just going to want to close right back up. So the single-stranded binding proteins are holding it open until we can lay down our new DNA. Can you see if right down here? Yeah. Okay. Holds strands apart. We'll write that. Holds the strands apart. Okay. Good. So let's label our directions. So this is double-stranded, right? So this is our 5' end of this top strand, and this is our 3' end of the top strand. Our bottom strand, because DNA goes anti-parallel, so different directions. So our bottom strand, this is our 5' end, and then we go to our 3' end of the bottom strand. Now, this is very important for you to know. We are always, always, always replicating in the 5' to 3' direction. So, that is, never forget that. That's probably going to be a test question at some point in your self-phys career. So, in order for us to start laying down DNA, we need some sort of free 3' OH end for our DNA polymerase to start working. So in order to do that, we actually lay down a little segment of RNA just to give us that free 3' OH end, and then it'll get removed later. So I'm going to use a little red dash for that. So, oh no. There we go. So that is our RNA primer, and the thing that adds that is primase. RNA primer added by primase, which is an enzyme that adds primer. Makes sense? Good. Okay, so this old strand is going in the 5' to 3' direction. So to make a new strand in the anti-parallel direction, it has to go 5' to 3' this way, our new strand. So we're adding, and we're always starting from the origin of replication and going out. So we're starting here on this side, and then we'll get to that side. But so this side has to go in the 5' to 3' direction this way for our new strand. So for our little red new strand here, that's, I'm going to draw it a little closer. That's our 5'. So this end would be 3', right? Because 5' to 3'. So that's our 3' OH end. And now our DNA polymerase can come in and start laying down new DNA strands attached to that. And we can keep going until we run out of DNA. Okay? So DNA primase, DNA polymerase did that. So that's our new DNA. with DNA polymerase. Okay, so that's our 5' end, and this is our 3' end. So we're going in opposite direction from the old strand. Good stuff. Okay, so this is our leading strand because we're going continuous, right? We're not stopping. So this is our leading strand. Okay, now let's tackle this side. So we still need to replicate overall in the 5' to 3' direction because that's just how it works, right? Because we're going antiparallel. So, but we have to replicate, like, as we're opening up more and more space for us to replicate on the old DNA, but we always still have to go in this direction. So we wait until it's open enough, and then we add an RNA primer, and then we synthesize in the 5', 3' direction. And then we open up a little more, we add another primer, synthesize in the 5', 3' direction. And again, primer, excuse me, 5', 3' direction. Okay, so all of these little chunks, this is our 5' end of this first chunk, 3', 5', 3', 5', 3'. Okay, so overall we're going in this 5' to 3' direction for our new strand, and because it's so like fragmented, so this is our lagging strand. And then each of these little chunks right here is called an Okazaki fragment. And then later, DNA ligase will come in and cut out all of these RNA chunks, not just in the lagging strand, but also in the leading strand. It's going to cut out all of the RNA, and then we're going to glue everything back together as one continuous DNA strand. And then we'll be like this example up here with just a continuous new DNA. So that was DNA ligase is going to come in, cut out the RNA, and glue everything back together. Cool. So everything is going to happen just like it did up here, down here, except opposite, because now this old strand, the 5', 3' is in this way, so we need to replicate this way overall. So this would be our leading strand over here. And then our lagging strand would be on this side. So same thing as up top, just opposite. Okay, just opposite directions, I mean. I think that's it. So we know that we always synthesize in the 5' to 3' direction. And another important thing to note is that this is called semi-conservative replication because we split it apart, and each new DNA is going to have one new strand and one old strand, one new strand, one old strand. So we're like half, so like semi-half, we're half conserving in each our old DNA. So semi-conservative replication, that might be important to know for tests at some point. I know he I think he likes that one. Who knows? But, okay. Why is this? There we go. Semi-conservative. Replication is what this is. Okay. That is all of that. Let's see, what else do we got? We'll do a little bit of transcription. So make sure you have that. I'm gonna erase it now. Okay. So if we start out, we'll write our DNA coding strand first. So just a random assortment that I made up. Sorry, I gotta look at my paper. Okay. G, C, T, A, C, A, G, G, T. So this is gonna be our DNA coding strand. And then if we make another strand of DNA from that, we would have to use the matching nucleotides, right? The matching bases. So it would be C, G, A, T, G, T, C, C, A. So this is our DNA template strand. Okay, so that's our DNA. If we want to, we could go in and code, we could do transcription. Sorry, one second. And then make RNA from this. So RNA will come from the DNA template strand. So, well, hold on, actually, let me write this out first. So this, to go from this strand, oh no, goodness. Okay, from this strand to this strand of DNA is replication. Okay, so if we're gonna make our RNA, then, okay, so it would be G, C, and then remember in RNA, we don't have any T's, we only have U's that match up with the A, so it'd be U, but we still have the A that would match up with the T there. C, A, G, G, U. So that would be our RNA transcript. So this here is transcription. And then if we want to use this series of this RNA code to make protein, we can do that. And I didn't think this far ahead, so just hold on one second while I bring up the amino acid chart. Okay, let's see. So this isn't going to make any sense. We're just going to come up with some random amino acids, but that is okay. Let's use pink. So when we read RNA to make protein, we read them in codons, so 3 letters at a time. So here's one codon, here's another codon, here's another codon. So each codon is going to code for 1 amino acid. So if you want, you can look in your notes and pull up a amino acid chart. He will not make you memorize this. If he wants you to go from RNA to protein, he will give you an amino acid chart in your test, which is nice. But if you don't know what I'm talking about, this might not work. There we go. That's what it looks like. That's just one example that I have. That will be in your past notes. But if you want to follow along, you can do that. Otherwise, just write it down. So if we look at our amino acid chart, our G, C, and U would match up to be alanine. So ALA, alanine. ACA would be threonine, so THR. And again, I'm looking at that amino acid chart to match up all three of these letters to come up with a codon that codes for a certain amino acid. So GGU would be glycine, GLY. Okay, so this is our polypeptide chain, or protein. These are all amino acids, right? So then this process here is our translation. So if you get confused what the difference between transcription and translation, this might help you a little bit. So when we are like hand copying and making a copy of something, but we're copying it like still in the same language, that's called like transcribing something. So the DNA and RNA still use the same type of bases. So it's still in like the same language. Otherwise, like even the U and T is still in the same language. We're just like using only U and RNA and only T and DNA. So that would be transcribing within the same language. But when we go from using our bases to using amino acids, that's like a totally different language. So we're translating from one language to the next. Okay, may or may not help you. That helps me. Important things to know that we haven't written down yet. So, sorry, I live on Brady Street, if you can hear that loud noise. Lovely motorcycles. Okay, so our start codon. You should definitely know what our start codon is. AUG. Remember that. Okay, and then our stop codons, you should also know. One of these would make a very easy test question, just would be good to know. So I like to write out UAA first, and then I just replace each A with a G one at a time. So, UAA, UAG, UGA. Start with UAA, replace one with a G, one at a time. So start codon, stop codon. So if I were to have actually drawn out like a full polypeptide, our first codon would have been AUG, and that would say to like actually start doing protein synthesis, and then at the end of our polypeptide chain would have been one of these stop codons, and that would say to stop synthesis. It would also be important to know that AUG actually does code for an amino acid. It codes for methionine. That is probably one of the only codons that he would ask you to remember what it codes for. All the rest, like glycine and threonine, he's not going to make you to remember all of these little codons. But AUG, coding for methionine, he would probably make you remember. The stop codons do not code for a codon. They just say they do not code for amino acid. They just say to stop protein synthesis. So overall, we have in our chart, we have 64 codons. That's a G, sorry. Okay, 60, oh my goodness. Okay, how do I draw 6? 64 codons. Wow, it's a little late, sorry guys. 64 codons. Oh man, so 64 codons overall, and then, but because these 3 codons don't actually code for an amino acid, we only have 61 codons that, that will code for an amino acid. I wish I knew how to like edit videos. I could edit out all of my little mistakes today, but you're just gonna have to bear with me. Okay, there we go. I think that's it. Maybe just to reiterate, like we were saying before, we're always gonna read in the 5' to 3' direction. So we're reading, like to make our codons, this would be the 5' direction, and this would be the 3' direction of this RNA transcript here. We're always reading in that direction to make our codons. 5', 3' direction. Yep. That's it. Email me or message me if you have any questions, and happy studying.